why.
Okay.
Okay, I'm here. Hi, Gavin.
How are you, Dick? Are you well?
Yes, I'm well. Thank you. You're spotlighted there with almost a halo around your head there, Gavin, by the way. Just yeah, no, I guess you and others, I mean, we're all kind of like shocked by the news about Paul Farmer, but
yes, gosh. Yeah, it just shows you, hey?
yeah, really. Take it
Has there
easy.
been any word what he died from?
I don't know. Obviously it was sudden and unexpected, but
Yeah, wow.
it was
Just shows you.
still working hard,
Yeah.
taking care of patients in Rwanda.
Yeah, incredible.
Yeah,
Anyway.
indeed. So Gavin, we'll just wait another minute in case maybe others had trouble connecting before we jump in. And yeah, as usual, it's kind of a bit of a mix of students and staff and people from elsewhere who are, in some way connected with us. So I'll start just by introducing you, Gavin. Honestly, a very brief introduction. I'm never very good at these, but I'm really pleased, honored to introduce Dr. Gavin Churchyard, who's been a real force in tuberculosis, particularly in randomized trials, and notably in TB prevention trials. He's the founder of the Orem Institute, which is a nonprofit, and I read the note. So not just research, but technical assistance and clinical service, and not just TB, HIV, but now COVID as well. And he also is the beneficiary or the winner, or whatever is the right word, of a recipient of a large grant from Unitaid to kind of find ways to scale up TB prevention efforts in a number of high burden, high incidence countries. So that's really what we were hoping to hear from you, is that experience and some insights. We've also been very interested here in the same issues. And yes, just as a last message before I let you take over, I would like to just sort of say a note that we again honor the memory of Paul Farmer, who died unexpectedly and suddenly in Rwanda on Monday at the age of 62. So on that note, I'll pass to Gavin, and away you go. Now, Gavin, are you going to show your slides, or do you need us to?
No, I'll show my slides.
Perfect.
Just let me do that. And I check that you can see my slides.
I can see them. They seem to be in slide sorter mode at the moment.
And now, can you see them in screen show now?
I still see them as a slide sorter mode.
All right. Well, I tell you what, I'm going to stop sharing and then put it into screen show and then share again. That's the problem. Okay. Oh, nope. Oh, you know what it is? It's because there's two screens. Okay, let me just give me a moment, Dick. I need to.
Sure.
Just see if I can. Now it's hung on the screen and it's not letting me. Okay. This should hopefully work. All right, now can you see it in screen show?
Yes, I can. Catalyzing scale up in big bold letters.
Great. Well, Dick, thanks so much for giving me this opportunity to present to you. It's a wonderful opportunity, and it's good to connect with some old colleagues. So what I'll be doing, if the slides will be advanced, just give some brief background and then talk mostly on impact for TB, as you mentioned, is this Unit 8 grant to scale up 3HP, but also look at some of the evidence for periodic 3HP and biomarker targeted 3HP, and if time permits, to talk a bit about MDR TB preventive treatment and future prospects, just that I'm involved with all those aspects as well. So starting with background, we're all aware that almost a quarter of the world's population is infected with TB and are at risk of progressing to TB disease. The WHO has made as a priority, based on the UN high-level meeting in 2018, the scale-up of TB preventive treatment amongst people with HIV, children less than 5, and then household contacts older than 5. And although we've made good progress in scaling up TB preventive treatment for people with HIV, we've made very poor progress for preventive treatment amongst household contacts, but particularly those older than the age of 5. And due to this, WHO convened earlier last year, or in the latter half of last year, I should say, a call to action 2.0 to try and reinvigorate the call to action to scale up TB preventive therapy, particularly amongst household contacts and those older than 5 years of age. So the WHO also, this is following the meeting we had at McGill, was it in 2019? I can't even remember. But part of what emerged from that meeting was identifying of research gaps. And just to, I'm not going to go through them in any detail, but just to say that part of the research gaps that were identified included barriers to implementation, and particularly access to diagnostics and drugs, the financing component, and understanding patient perspectives, and then identifying research priorities along the cascade of prevention. And just to highlight here, the issue around initiating TPT and new products for preventive treatment. And then also the importance of identifying high-risk individuals to target TB preventive therapy at. And I will be focusing on the 3 areas: people with HIV and household contacts less than 5 or 5 years or older. And as I will hopefully demonstrate, the Impact for TB project addresses many of these research priorities that were set. So just by way as a reminder, 3HP is of course shorter than 6 to 12 months of isoniazid preventive therapy. It's less toxic, it's associated with better adherence, and it has a higher barrier to resistance and similar efficacy in HIV uninfected and infected persons, including children, and is less burden to the health programs. And this was demonstrated in study 26 done in the US, Canada, Brazil, and Spain, and then as well as in a high burden setting, South Africa, where 3HP was shown to be similar to 6 months of isoniazid, as well as continuous isoniazid preventive therapy. 3HP, this is modeling done by David Dowdy, and I was part of this paper where we modeled the cost effectiveness of 3HP compared to 6 months of isoniazid in used Uganda as an example. And what we showed was that if the price of rifapentine could be reduced from its initial price of $72 per patient course down to about $15 per patient course, then it may become cost effective under certain circumstances. So now moving on to the Impact for TB project, which was funded by Unit 8. This is a $59 million grant. And we were funded to do market shaping and the supply side chain work, which is really looking at the products, the formulations, and addressing those gaps and helping reduce the price of rifapentine. Then focusing on delivery of 3HP, specifically with respect to people with HIV and children less than 5, and then generating evidence for policy. So this was to address policy gaps, and the evidence will emerge from here. This was funded for 4 years. We will be reaching the end of those 4 years in August of this year, but we have now got as I'll talk to you later, no cost extension, and following that, a costed extension. So the goals are to reduce TB incidence and deaths amongst people with HIV and child contacts through sustainable implementation of quality assured, affordable 3HP. And this is done by increasing the number starting 3HP in these risk groups. We have, these are our consortium partners working with us. We are working in 12 countries, which include low income, low middle, and high middle income countries. So this just in a figure outlines the activities of the Impact for TB grant, divided into the market shaping, the delivery, and generating evidence, and then supporting WHO for global scale-up. So starting with the market intervention aspects. So I've already mentioned, well, sorry, I'll come to the price. So we have been hard at work to increase the number of formulations, starting with development of a fixed dose combination, isoniazid and rifapentine. And so we now have the isoniazid single tablets, the isoniazid single tablets from the originator, that's Sanofi, which is a 120 milligram tablet. And that's this, sorry, 150 milligram tablet, sorry. But that's as a single tablet. And we now have the fixed dose combination of 300 of isoniazid and rifapentine. This is manufactured by McLeod's. And so when we look at the products that we currently have available, this is the originator shown here, the rifapentine 150 milligrams. But this requires that adults take 6 of the rifapentine singles and 3 of the INH plus 1 of pyridoxine, so they take a total of 10 tablets. We did reduce the price starting initially at 72, and even before the project started, we had reduced the price down to $45 per patient course. And then with the intervention of the project, we reduced the price of the original rifapentine down to $15 per patient course. But this has subsequently gone up because of issues around that I'll come to now with the impurities and the scale-up not being as fast as what Sanofi was hoping for. We now have the fixed dose combination, as I mentioned. This is produced by McLeod's. This is currently available, and the Lupin equivalent of the FDC will be available later this year. This is currently priced at $15 per patient course, so it's actually cheaper than the original rifapentine combined with isoniazid. And we are hopeful that there will be further price reductions as we scale up. So McLeod's are increasing their production capacity to greater than 2 million patient courses annually, and Lupin will be gradually increasing theirs also to 2 million patient courses based on demand. Just to mention that the Sanofi was really only looking at 400,000 patient courses, which is what we were contracted to do under Impact for TB. So we were aiming to catalyze a total of 400,000 people starting on 3HP during the course of the Impact for TB project. That was our goal. But so we've got an FDC. The 150 milligram tablet is a bit challenging in that you need to take many tablets, and it introduces problems now as we're moving into 1HP, as well as the rifapentine, moxifloxacin 4-month TB treatment regimens. And so there was a need to come up with another single formulation, which is the 300 milligram a single tablet, which Lupin is now developing. And they will also scale up the production of the single tablet. So now I'm looking at what's going to emerge as these new formulations come on board, as the Lupin single and FDC comes on board. So currently, as I've said, but you take 10 tablets for 3 HP currently and 6 tablets for 1 HP. But with the FDC now coming on board, you can take 4 tablets for 3 HP as well as 4 tablets for 1 HP. But as soon as we get the 300 milligram single, it will now be possible to reduce the pill burden for 1 HP down to just 3 tablets compared to 4 for 3 HP. So the pill burden has come down quite dramatically from 10 originally. For 3 HP, it will come down to 4, and for 1 HP down to 3. All right, so that's for adults, but what about children older than 2? Don't forget, we don't have the PK data yet for children less than 2. And so there are these weight-banded dosage regimens. But of course, this is, again, problems because we're going to have to use the Sanofi 150 coated single tablet, which means that children, very, very young children, will battle to swallow that, which means they will have to crush the tablets. And currently, we don't have any data from crushing studies on the bioavailability of the crushing. So these are issues we're going to have to further address. So it's really important that we do specifically address the formulations for children because there are many childhood cases of TB that we need to prevent, both cases and deaths, and this is an effective way of averting that. So a group convened by WHO, but WHO, PADA, TB, GAP-F, and ourselves met and discussed the ideal formulation for the pediatric tablet. And here we're looking at a fully dispersible tablet that is functionally scored. And so the aim is to have a 150 milligram tablet, but now that it will be functionally scored, that means when you break it, it will be exactly half, rather than just often what you see like the McLeod's FDC is scored, but it's not functionally scored. If you break it, you won't get exactly half the dose. And so that has been deemed to be the ideal dose and formulation for children. And it's now on the expression of interest list at WHO. And this is now moving forward. I'll come back to an FDC for children when I discuss the trials in children a bit later. So one of the challenges we've had is an impurity in rifapentine. So nitrosamines are present in food, drinking water, tobacco smoke, and these are carcinogens with increased risk of cancer after decades of exposure. And these nitrosamine impurities were discovered in the rifamycins. It's not unique to rifapentine, so rifampicin, rifapentine, and other medicines. And the plan was to reduce the nitrosamine impurities down to below 20 parts per million. And this was agreed to by both FDA and WHO. And the suppliers are working to reduce the levels down to as low as 0.1, although we're not entirely sure that that is actually achievable from a chemical processing perspective. With respect to the pediatric formulations, the good news is that the same limit was set, but the current pediatric formulations are well below 20 parts per million. But we do need to just bear in mind that the risk of not treating or preventing TB far outweigh the theoretical risk of cancer from taking an incredibly short dose of rifapentine. So now moving on to the delivery aspects of impact for TB. So this slide is from the middle of last year, and what it showed was that we were still in the process of scaling up 3HP, both being delivered and starting to implement the 3HP scale-up. And shown on the right is the timescale for implementing this. I'm now glad to say, apart from Tanzania, which we had to drop, there were huge challenges with the Ministry of Health and strong opposition to implementing in Tanzania initially. We're still working with them, and hopefully we are getting closer to changing that policy. But in all other countries, we are now implementing 3HP, although in some not quite at the scale that we had hoped, including South Africa, but also India, for instance. So moving on to catalyzing global scale-up. So we were, just to remind you, we were tasked with starting 400,000 people on 3HP, both within our project countries, but also that included the global scale-up. And so what was decided was that the ordering and distribution of 3HP would be done through the Global Fund Antiretroviral Therapy Technical Working Group. And people who sit on that are the PEPFAR groups, of course, Global Fund, other partners, and then WHO, PAHO, Gates Foundation, etc., USAID. And so all the ordering has been coordinated through this technical working group. GDF also sits on this technical working group. And I'm glad to say that we were tasked to scale up, catalyze 400,000. During the course of last year, we catalyzed the ordering of 1.8 million patient courses. So a quite dramatic uptake of 3HP. And it's been undergoing these quality checks, because before they can be distributed, we need to check the level of nitrosamines in the product that will be distributed. And this is being done by FHR 360. And 1 million of that is now being delivered and is being rolled out. So that's really very encouraging. And these are the countries that have placed orders either for the rifapentine single, the FDC, or both. So there's 34 countries currently that have placed these orders. And then in terms of how we've had to do this, because quite honestly, the supply of 3HP was rapidly outstripped because of how successful we were in catalyzing uptake. So we've had to phase the delivery and scale up of 3HP. And in this map shows you the sequencing of the scale up by countries. And so it will be done in tiers over, starting last year and continuing this year and into 2023. But in terms of the FDC, we now have 5 countries that have already registered the FDC, and it's under review in 20 other countries. So the FDC is also, the uptake is also quite promising, and we hopefully will transition largely into FDC. So now moving on to generating evidence. So these are the projects that we're doing in Impact for TB to generate evidence for policy. So the Dolphin trial has been completed, and here we looked at people with HIV who were on efavirenz-based antiretroviral therapy, who was switched to dolutegravir and a washout period of 2 months was allowed before we initiated 3HP and then did intensive and sparse PK sampling. And what the study showed was that there was no need to double the dose of dolutegravir amongst individuals that are unstable dolutegravir. Dolphin 2 is currently enrolling, and this extends that study where we're looking at ARV naive individuals starting dolutegravir-based antiretroviral therapy simultaneously with 3HP and looking at viral loads. And what we are hoping to show is that, again, it will be fine to start 3HP and dolutegravir-based ART simultaneously, and that there's no untoward impact on viral loads. We are evaluating models to help scale up delivery, and the first is choice architecture-based preventive treatment. This has been led by Chris Hoffman, and here this is a study to evaluate opting out. So 3HP is prescribed in people with HIV, unless they opt out of it, either the physician or the patient. And so we are looking to see whether this will dramatically increase the scale up of 3HP or the uptake of 3HP. And this is currently ongoing. We've now moved into, so let me just go back. We've now just moved into the era of 1HP. So great, 1 month of treatment daily now of rifapentine and isoniazid. It's non-inferior to 9 months of isoniazid, but there are challenges to this in that the cost of rifapentine is substantially more. And so we've already done a cost effectiveness modeling, and again, 1HP may become cost effective relative to 3HP if we can get the price down to $15 or below, which we've already done. But it's also very dependent on the prevalence of the TB infection, efficacy, and the cost, as I mentioned, and importantly, the completion rates, which we anticipate will be even higher than for 3HP. So we now have regimens we can choose from: 3HP, 1HP. So taking treatment once a week for 3 months versus daily for 1 month. So what will be the preference of patients and providers, and what will be treatment completion rates, which we think will be the important differentiator. And so the 1-2-3 study is comparing 1HP to 3HP. This is amongst people with HIV as well as household contacts. And this will be done in South Africa, Indonesia, India, and Mozambique. And this has just undergone review by WHO ethics. So we'll be starting in the 3rd quarter of this year. So one of the things we also looked at during this project was the dosing of rifapentine. There was data that came out of Rada Savage group showing that rifapentine metabolism was not dependent on weight, and that one could do flat dosing. And so that information has been fed into WHO. We now recommend flat-based dosing for 3HP and for 1HP. And this has important implications for the new rifapentine moxi TB treatment regimen. So another study that we're doing to try and improve the uptake is comparing community versus facility-based child contact investigations. This has been done in 32 clinics in South Africa and Ethiopia, and this study is currently underway and includes both the quantitative and qualitative aspects. So pediatric TPT, particularly in the household, is challenging because depending on the age, whether the child is HIV infected or not, and depending on what's available in the country, one may need to use 3 different TPT regimens for children. Now, that's not ideal. We want 1 TPT regimen we can use for the entire household, which is what we're aiming to achieve with 3HP. So there is work to do this, and we are co-funding the TBTC study 35, which is looking at the PK of 3HP in children less than 12 years of age. And this is using the FDC. So this FDC was available from Sanofi, but they only manufactured it for this study, and we weren't certain whether they were going to continue. In fact, they told us they weren't. And so the next study is the Dolphin Kids study, which is under Impact for TB as well, where we're assessing the safety, tolerability, and PK of 3HP in infants and children and adolescents taking a dolutegravir-based ART. And we thought we were going to have to use crushed singles, but we just heard this week that Sanofi may be making another batch for us to use in these studies. So that's very assuring. So what about Tptn Pregnancy? So the Aspire trial, which used IPT in pregnancy, funded by NIH and led by Mita Gupta, showed that there were adverse pregnancy outcomes, and these findings were replicated by women who became pregnant while taking IPT in the brief TB trial, which was the 1HP versus 9H study, again showing very similar findings of adverse pregnancy outcomes. So what's about 3HP? So the IMPACT 2001 study evaluated rifapentine in pregnant women and showed that there was no need to alter the dose of rifapentine in pregnant women, and presented at the Union Conference last year. This was from our WIP3TB trial, that a pregnant woman on 3HP, woman on 3HP who fell pregnant had similar adverse pregnancy outcomes, whether they were exposed or not exposed. But to further validate this, we are going to be doing the Dolphin mom study, also again under IMPACT for TB, to evaluate safety, tolerability, and PK, comparing 1HP versus 3HP, initiated both antepartum and postpartum. So this is the IMPACT for TB timeline. I've already mentioned that we've got a no-cost extension and a costed extension. And in the costed extension, we're going to be scaling up these second generic products of the 300 milligram single and the FDC. We're going to be looking at generating evidence for 1HP versus 3HP, and that will be done through the 123 study. But also we are donating products to countries that have expressed interest in scaling up 1HP, and we have identified some countries to do that. And then we will complete these studies that I've just mentioned. All right, so then moving on to periodic 3HP. So this was the WIP3TB trial, which I led and was funded through the Challenge TB grant and US aid, that compared the effectiveness of 3HP given annually versus once in people with HIV. But the first part of the study compared the treatment completion of 3HP to 6 months of isoniazid. And what we showed was much higher treatment completion of 3HP versus 6H. And in fact, a lot of people were skeptical of this result, but David Dowdy, who's part of that study, discrete choice experiment study in Uganda, has shown even higher treatment completion rates than what we showed here. They showed 94% with self-supervised 3HP. So it does look like in high-burden countries, 3HP is associated with high treatment completion rates. Then we compared the 3HP given once versus annually to see if we could kind of find a better way of ensuring sustained protection instead of giving continuous, lifelong, daily isoniazid. And what we showed was that amongst people on antiretroviral therapy, that giving an additional course of 3HP did not give any additional benefits. The TB incidence was the same, death was the same as well. And in all subpopulations, there was no difference in the effectiveness. So what about biomarker-targeted 3HP? So we know that there have been biomarkers that have been identified. There's a whole range of them now, and they identify people at increased risk of developing TB, and they're meant to be markers of incipient TB. The risk 11, which was formerly known as the core or the correlate, was one of them developed or identified by the SATV group. It's now called risk 11. And what they showed in the Cortis trial was that this performed very well as a diagnostic. The diagnostic risk ratio was 5.1, and it also identified individuals at increased risk of TB with a cumulative incidence ratio of 2.6. And it had similar performance in people with HIV to those without HIV. But when the study also then evaluated giving 3HP to those that were risk 11 positive, and so people were randomized to, it was a randomization where it was people that were risk positive got it, and then people that were risk negative were randomized to either get treatment or not get it. And what we showed was that in the intention to treat analysis, there was no difference. And in the per protocol analysis, again, no difference, but the treatment efficacy was estimated at 22%. In the per protocol analysis, just to point out that in the people who took the 3HP, that there was 9 months where no one got TB. So this may have been partly because of the initial screening, although we didn't see that in the other group, but that there may have been limited duration of protection, which was lost due to reinfection or inadequately treated infection that then rebounded. So either this correlate did not identify people who would benefit from 3HP, or 3HP is inadequate to identify people that have a positive correlate. So, Dick, should I continue, or do you want me to just progress to the conclusions?
Well, I mean, you're doing, I mean, it's 1:36 according to my clock. So I think there's certainly some time, and we'll still have plenty of time for questions. You think you'll be able to, yeah, go ahead.
Let me do it quickly. So moving on to TB preventive treatment for MDR TB infection. So modeling shows that although TB incidence overall globally is declining, albeit slowly, the infection incidence is increasing. The prevalence of infection is increasing in all WHO regions. So this is a concern, particularly in children. What we did in the Phoenix trial, which is the large trial that I'm leading, funded by NIH and being done by ACTG and Impact Networks, in the feasibility study preceding that, we enrolled household contacts of patients, index patients with rifampicin-resistant TB, and showed that a high proportion were at high risk of developing TB, 77%. And of those that had no risk factors at enrollment, so those were people that were older than 5 and HIV uninfected and didn't have evidence of infection, 20% or 21% of them over a period of a year became infected. So that suggested that part of them, some of them may have been infected from the community, but it's also likely that individuals were in the process of becoming converted and subsequently converted their quantiferin and TSTs post us first seeing them. So when you put this together, the vast majority of household contacts are, would be at high risk of developing TB in MDR TB exposed households. TB incidence is highest in children less than 5 and HIV infected, but even those that are HIV infected older than 5, this incidence of almost 2% per year is similar to what we now see in people with HIV on antiretroviral therapy. There's 3 trials, as I'm sure you're all aware, that are currently evaluating this. 2 evaluating levofloxacin against placebo, so that's TB Champ and B-Quin. Both are going to be reading out hopefully this year, so we should get some interesting data. Phoenix is still enrolling, was severely impacted by COVID and some other challenges, but we're getting back on track, and that we'll only get results the earliest 2026. And that's evaluating delaminate versus isoniazid for MDR TB. But that's not the end of innovation. There is plenty of innovation that we can look forward to. And so WHO has put out target product profiles for TB preventive therapy, identifying both priority attributes and the attribute characteristics. And the modeling suggests that in a country like South Africa, if we had a new regimen that met the optimal criteria and that was scaled up, it could potentially have a population level impact, quite substantial population level impact. But thinking through of new regimens, I can't talk to you and not mention Dick's study of the 2R2 study looking at the 2 months of high dose rifampicin. So that's, I don't know where you are in that study deck, but I mean, I think that's going to be really important and will further add to ultra short course regimens. And the other is the asteroid study evaluating 6 weeks of rifapentine alone compared to other short course rifamycin-based regimens. But of mice and men, the mouse model that identified 6 months of isoniazid has been effective, 3-HP has been effective, and shown here, 1-HP has been effective. So the mouse model has been replicated in clinical trials, phase 3 clinical trials for 6H, 3-HP, and 1-HP. The same mouse model identifies that giving both a combination of rifapentine with bedaquiline, and even though rifapentine will induce metabolism of bedaquiline, the PK modeling suggests that we should still see a substantial effect, and we could potentially reduce treatment down to 2 weeks and possibly even as low as 1 week. In terms of the, and that's, so you can see that's substantially less than the 1-HP regimen shown on the highlighted on the left. But for MDR-TB, if we use bedaquiline alone, given just 1 month of treatment, has similar efficacy to 1-HP. And so that's on the CFU mouse data, but the relapse data suggests that you may need 3 months of bedaquiline. But if you couple bedaquiline with other drugs, and so I already mentioned rifapentine, but in another mouse study, combining it with drugs like delamanid and sutezolid increases its potency and again could substantially reduce the treatment duration. But giving long-acting formulations of bedaquiline have now been developed, and the mouse data suggests that we could actually, giving a single injection of this long-acting bedaquiline has the same efficacy or similar efficacy as giving 3 doses 1 month apart and giving bedaquiline orally for 3 months. And so this has become an area of development that we are focusing on. There are also long-acting injectables being developed for rifapentine and for isoniazid, which will enable us to give the 2 injections, much like we do now for HIV prevention. And so TB preventive therapy may become a single injection. And so it will remove all the adherence issues, does introduce other complexities, but nevertheless, I think we've got some exciting developments that we can look forward to in the coming years. So in conclusion, we need to manage this global burden of infection, and it's essential to meeting the NTB targets. The rifapentine-based short-course TPT is associated with better adherence and safety, and may be given with dolutegravir-based ART. And scale-up of 3-HP is ongoing, but hampered by the cost, the impurities, and the lack of child-friendly formulations. And a single round of short-course treatment is effective in TB high-burden countries. And further innovations are required to improve potency and further reduce the duration of treatment for both drug-susceptible and drug-resistant TB, and to maximize the impact on health. And ultimately, there is the prospect that we may have a pan-TPT regimen through this innovation. So thank you very much.
Great, great. Okay, thank you, Gavin. That was great. A real tour de force with, you know, one major study per 30 seconds shown there. So I'd imagine people are still trying to absorb all that. But so we'll see if anyone has questions. Marcel has a question, but we traditionally try to let the students go first before we let Marcel speak. I have a few as well. Anybody want to jump in first? Or we'll let, oh, Marcel, why don't you go ahead while we're waiting for students? And if anyone, oh, sorry, actually, go ahead.
Hello, I wanted to understand about the protection one could get from TB after completing the treatment of 3HP. Approximate number of years in if there are any papers on that already available. Thank you so much.
All right, thank you. I'm just going to quickly share my screen again so I can show
Sure.
you later. Let's see.
So duration of benefit, is that it?
It's duration of benefits. And we can look at two studies. Oh, no, that's in the wrong view. But anyway, let me not waste time. So just let me come here to the initial 3HP studies, and I showed you the study 26, and then the one from South Africa in Soweto. Sorry. And the study in Soweto here, you can see, so it was true for all regimens, but that the protection was quite limited. That if you look at this in more detail, and particularly in the per protocol analysis, that the duration of protection in this setting were in an era when antiretroviral therapy was being scaled up. So at the start of the study, very few people were on ARVs, and by the end of the study, about half were on ARVs. The duration of protection was about a year. In my Tbeta TB study, very, very high rates of transmission in the South African gold mines. The moment you stop taking TPT, it immediately rebounded. So there was hardly any duration of protection. But both the scale up of antiretroviral therapy now is quite dramatic. So that's really great. And with the scale up of TPT, we've seen a changing in epidemiology. And when we look at the results of the 1HP study that I showed you here, so just focusing on this graph. So this was done in 52 clinical trial sites in 12 countries around the world, including in very high transmission settings. So we're having a thunderstorm at the moment, but including in South Africa. There were 6 sites in South Africa. And true in South Africa as in all other sites, that these individuals took 1 month of treatment or 9 months of isoniazid, but the benefits lasted for 5 years. So there's been quite a fundamental change, and it's likely due to the high uptake of antiretroviral therapy and declining rates of TB in these very high rates, high transmission settings. And so, and because our WIP3 TB trial also showed that there was no benefit of taking a second round, where if those rates were very high, if the durability was being lost, we would have expected to see a benefit, and we didn't see that. So I think there is some evidence to suggest that taking a single round, depending on epidemiological setting, may be adequate.
Right. I could just add that the follow-up study to the Temprano study, 7-year follow-up, and also I forget how long, but a long follow-up in the Rio study, both concluded that there was evidence of sustained benefit 5 to 7 years after, in settings with much lower rates of TB. Although I think even in populations where the ARV uptake was not as high, but much lower rates of TB than in South Africa. Okay, so Marcel, do you want to?
Hi, Gavin. Thank you for zooming over to join us. I have a sort of a more conceptual question. As we know, when the preclinical models are excellent, such as COVID, and the vaccine works in the preclinical models, we can have success in the field. We have TB models of protection by BCG, which probably looks like what it does in people. And we have TB models of disease. Do you think that we have adequate models of tuberculosis infection to inform the next interventions? Or are we sort of just iteratively doing kind of, a trial and error with the tools we have?
Thanks, Marcel. So unlike, I'm not sure I would agree with TB vaccines that the mouse models a good model, or even the NHP model is that great. But there is more. Well, we have some evidence there. Treatment, the mouse model is pretty good. We really have a good understanding of how it works, and there's all this translational modeling to inform TB treatment now. For preventive treatments, unfortunately, we don't have any biomarkers that will enable us to do phase 2, phase 2A and 2B trials. So all we have is the mouse model, and we've got to leap straight into doing the efficacy trials. But thankfully, the evidence suggests, as I mentioned, the mouse model that was developed by Eric and by Jacques Rosset seemed to be really quite predictive of the efficacy observed in humans so far. We haven't had a failure, and so that is continued to be used to inform as we go forward. So, for instance, the long-acting injectables are first being tested in mice, and so and based on that, the move into clinical trials. So it's unfortunately the only model we have, and we can't stage gates and moving into sort of efficacy trials. So you take the mouse data, you make a decision, and off you go. And if it's a mistake, it's quite an expensive mistake, unfortunately.
Yeah, I don't know if you've read the details, Gavin. I recently read them with Dick and Andrienne, if she's on. And the model actually uses a recombinant BCG that nobody has as an aerosol pre-challenge before a high-dose MTB. So it may be predictive, but it's not what you would call a very portable model, unfortunately.
Now, that I would agree with, absolutely. Yeah, it's specific to Eric. So if Eric's model predicts it, then that's probably right.
As long as it predicts human events, it's better than a model that doesn't. Thank you, Gavin.
Thanks, Marcel.
Does anyone else have a question? I have a couple of questions, but I don't want to, hog the question period. One thing I was curious, Gavin, is the most of your studies have been in under 5 and people living with HIV. If I got it right, have you been doing more studies in the kind of older non-HIV infected household contacts?
Yeah, so that is what the 1, 2, 3 study is doing. It's looking at from people 13 years or older. So they will be both HIV infected and HIV uninfected. So people in care, people with HIV in care, and then household contacts, regardless of whether they're HIV infected or not. But that's the older household group, household contacts, because I agree they're quite neglected.
Okay, and in those the other thing was, what about testing for TB infection, whether it's IGRA or tuberculin skin testing and chest x-rays? Have you been incorporating that in your scale-up efforts, or?
So we have, we promoted the scale-up of 3HP. We help countries, and quite honestly, it's whatever they have in their policy. And currently, it's just symptom-based screening for both people with HIV and household contacts. So access to X-rays is increasing, but it's definitely not ubiquitous, and it's really not policy anywhere. It's not sort of you have to have an X-ray. And we now know from the WHO TB screening guidelines that meta-analysis showing that amongst people with HIV on ARBs, that X-ray substantially increases the yield of TB cases detected. And so we really need to encourage countries to not just do symptom-based screening, but also chest X-ray screening. In the clinical trials we're doing, we do both symptom and chest X-ray. But programmatically, we haven't pushed chest X-ray. Unit 8 felt they wanted us just to focus on the treatment component, not on the screening aspects.
Okay, and are you monitoring, and again, in these larger scale-up kind of projects, monitoring drug resistance?
Yeah, so it's something that we've got I didn't actually put it into my presentation, but in the 12 countries, we have sentinel sites. So we have at least one. So I think on average, we've got 2 or 3 sentinel sites per country. And that's where we are taking people enrolled into the routine program. We're tracking them through that cascade of prevention and post-treatment, so looking for occurrence of TB, and if they have, then whether that's resistance or not. So that's been done through our sentinel surveys. Then in addition to that, again, I didn't have it in my presentation, but we have a study funded by the Gates Foundation called Opt for TPT, and there we are doing it in looking in South Africa, Zimbabwe, and Ethiopia. Again, very similarly, having sentinel sites looking at in detail the scale up of 3HP as well as 6H in these different countries. So from those sentinel sites, we will be collecting information on drug resistance. Then, of course, in our clinical trials, we're also collecting it.
Right. No, but in the clinical trials, once you've done chest X-ray screening, presumably you've eliminated a fair number of people with active TB,
yes,
at least the most advanced. And so hopefully you've reduced your risk of drug resistance, but it's in the groups where they're not doing chest X-ray that you might be more worried about generating drug resistance.
Yeah, that's correct.
Yeah. Okay. Okay, that's interesting. Anyone else? Really,
There's a question about India. So India is one of our countries. They are doing implementation study at the moment, but not in all the sites. But they are planning to scale up 3HP. And of course, both Lupin and McLeod's are based in India. So they certainly support. It's just taking longer than what we had anticipated. But that will be for both people with HIV and household contacts.
okay. So I don't know if Jonathan, if you wanted to say out loud your question.
Yeah, I can say it out loud. It was more, thought the boat had passed after the first conversation. So it was more just kind of looking at the data that was presented and the question posed, that we're pretty confident that all TPT regimens that are recommended are pretty good and effective against your current infection with M. tuberculosis, but we're uncertain on duration of protection offered in the context of re-challenge, because that's pretty consistently what I've seen, and then that perhaps the longer duration of protection we're seeing now, as you say, is due to ART. So you have a more immunocompetent population that might just be more resistant to reinfection. So I just wanted to know your thoughts, or if that was a fair interpretation of the data as presented.
It's a combination of ARV plus TPT. It's not ARV alone. I mean, the Temprano study showed very clearly the benefit of giving both and giving it early. And plus, we've got other data from the Cape Town study, observational data showing that benefit. But now that ART is very widespread, the benefit of TPT is becoming much more durable. But of course, it doesn't preclude someone who has been effectively treated from being reinfected. And so if we had a test that we could use to identify people who were effectively cured of the infection and who then became reinfected, we could then target retreatment to people who have become reinfected. Because we know that recent infection is associated with a very high risk of progressing to TB disease. So we don't just need new treatment, we also need better diagnostics to help us identify those who would benefit.
Right, and can I just ask one follow-up on study 26? I think that enrolled patients in the US and a large number were contacts. So I'm like, in the non-HIV population, has there been like a stratified analysis on durability of protection among people in a low incidence setting like the US versus the high incidence setting that perhaps I'm unaware of?
I'm not aware of it. They certainly
Okay.
they enrolled additional people, people with HIV into that trial, as well as children, etc., and showed, replicated the results. But I'm not, that study, it still have quite a long follow-up though, but I don't know if it's been sort of looked at more closely. Dick, are you aware?
No, I'm not aware of any kind of analysis within a single study. Really, it's the only way you get at it is comparing, like your the Tabella study where you had the highest incidence of TB of any study I've seen, and the duration of protection was the shortest, it seemed, and there's sort of a very rough correlation between incidence and duration of protection.
Yeah.
When you look across studies, again, in largely ARV naive HIV infected people.
Yeah. So there is a study, so both in Tabella, we looked at incidence by time period of follow-up, and the same person from the London school also did it in some of the other studies. I'd have to find it for you. But there is one other study I'm aware of.
Okay, thank you so much, and thanks for the great talk.
Great. Okay, so unless there's any other burning questions. So Gavin, yeah, thank you, really. It was great discussion, very impressive as always. Series of well-integrated projects tackling, a whole host of problems, from the very pragmatic, getting the drugs in the country to the more scientific of new predictors and that sort of thing, and biomarkers. So really, thank you very much. Very inspiring, and we'll look forward to further New England publications, please.
Thanks very much, everyone. Bye-bye.
Bye-bye.
Bye. Oh, I see my camera was off. I didn't realize. Oh, how long has my camera been off for?
Since you gave the talk, there you are.
I'm sorry. Apologies. I didn't realize that it was off. I was too engrossed speaking. Apologies about that.
No problem
But
at all.
thanks very much, Dick, and
Thank
everyone.
you.
Thanks,
Bye-bye.